An introduction and overview of cytokines and chemokines. Because cytokines and chemokines play such an important role in all parts of the immune response, this presentation will focus on all 5 major themes of this course. Now, at the end of this presentation, you should be able to describe the major roles of cytokines and chemokines in the immune system. You should be able to describe the 3 major modes by which cytokines and chemokines act on cells, and describe the major families, structural or functional families, of cytokines and chemokines. What are cytokines and chemokines? These are small proteins that bind to specific receptors on target cells. As shown here to the left, the example of interleukin-7, binding to receptor chains that are in the membrane, sending signals to intracellular signaling molecules that ultimately lead to changes in gene transcription. This is how cytokines and chemokines work. They change cell behavior by altering the function of proteins in the cell and by changing the expression of specific genes in the cell. Different cytokines have differing effects on cells. Some of these effects oppose one another. Some cytokines turn responses up, and some cytokines turn responses down. So what's the difference between cytokines and chemokines? Well, cytokines direct the growth, development, maturation, activation, and the lifespan of immune cells, whereas chemokines, which were called chemokines because they're chemotactic cytokines, they direct the movement of white blood cells in the body. These chemokines tell white blood cells where they should go. The expression of cytokines and chemokines and their receptors is highly regulated in the immune system, and some cytokines and some receptors are expressed on T cells and some on B cells, but they're not necessarily the same ones, and depending on the type that are expressed on the different types of cells, that dictates how the cells respond to these various signals. Now, cytokines and chemokines have 3 major mechanisms of action. So to the left, you see again the mechanism that they're following an activating stimulus. A cell is able to make cytokines. Those cytokines are secreted, and they act on a receptor that is present on a cell, on a target cell. These, when the cytokines act on that receptor, they send signals inside the cell to change the function of proteins and regulate gene transcription, and that leads to biological effects. Now, the 3 modes of actions of cytokines are, number 1, autocrine. That's where a cell gets activated, it secretes cytokine, and that cytokine feeds back on the same cell to stimulate itself. The second mode of action is paracrine action, where an activated cell makes cytokine that goes out and activates a nearby cell. And the third mode of action is endocrine action, where the activated cell makes cytokine that gets secreted into the circulation, and that may activate a cell that's far away from the cell that's actually making the cytokine. So these 3 modes of action, autocrine, paracrine, and endocrine, are all utilized by immune cells as they secrete cytokines and chemokines to increase or change their function. So there's a couple of ways to group cytokines and to think about them. One way that people lump cytokines together is by structural grouping. And as you can see on the left-hand side, these are the structural groups of cytokines: interferons, tumor necrosis factor family, interleukins, transforming growth factor beta family, colony stimulating factors, etc. On the right-hand side are the chemokine families, the C, the CC, CXC, CX3C chemokines. They're named this way because of their structural similarities that are shown at the bottom. For me, I have difficulty remembering the cytokines and chemokines based on their structures, and it's much easier to remember them based on a functional grouping approach.